I decided to move my laboratory down to Australia and down to Monash. I was very lucky that I've been able to convince 6 of my staff to fly all the way down from Northern Europe to Melbourne with me, which really helped in setting up the laboratory because I had people that knew what they were doing and we could really hit the ground running.
Roger's come to us from Copenhagen and he's brought with him a really highly functional, highly effective research team. It was really important for this team to be supported by veski. It really was a demonstration to them that they're going to move their lives from Copenhagen to Victoria, and again this commitment from Victoria to the research that they're undertaking there.
My particular interest is the brain, and I'm interested in understanding how the brain forms and how it functions. So if you think about the human brain, it has around 86 billion neurons. Massive number of cells, very, very complicated. But the worm nervous system, in contrast, has only 302 neurons. And even more interesting, that the genes that control the development of these cells in the brain and in the whole organism are very highly conserved to humans. So what we're trying to do is understand how these genes control brain development and function, and use that information to form hypotheses as to their function in humans. We actually use a jellyfish protein called green fluorescent protein. So we place this protein in the nervous system, for example, and that enables us to follow the development of these neurons. We can watch them migrating.
Those discoveries of how neurons function, how they interact with their cells that are the neighbouring cells that are important for their development, will provide absolutely new approaches to investigating neuronal function in mammalian systems.
So we're working on a particular microRNA that's been shown to be associated with schizophrenia in humans.
With a field and background in developmental neuroscience, how the nervous system develops. And that was something we were wanting to build here at Monash, and getting somebody really of Roger's background, experience and pedigree was something that we were very excited about.
I went to a top neuroscience lab at Columbia University Medical Center. There I made some really interesting discoveries. I was working on how the brain and how the nervous system responds to environmental stress. And it's only been 7 years, and now the discovery I made is being used in the clinic to help premature babies. It's crucial to engage young people in science. These are the people that are doing the work, and these are the people that are going to take things forward in the future. I've worked in Europe quite extensively and also in the States. And what I've noticed when I've arrived here, there's a real support network that I hadn't necessarily had in other locations. And that really stabilizes you and enables you to hit the ground running, and that's really what I want to do. We're in a very competitive area of research, and there's no prizes for getting second. So we really need to push our research forward, and the support of the Victorian government and veski in particular is crucial for that.
The real advantages of being able to bring a person such as Roger to Melbourne and to Victoria is to raise the profile of the research, to raise the quality of the research, to act as a magnet for other researchers we're trying to get.
So we're really at the ground level, in the trenches, trying to find out how these genes are working, and we're going to use that information with industry links in the future to hopefully identify potential drug targets.